to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 65th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I have been in the LBA and cell and gene therapy space for over 29 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 29 years too.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas, John is in upstate New York, and our producer is in Missouri, so we're all over the map covering the U.S. for you. We are thrilled to have you listening to the 65th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of your choosing before discussing any feedback we've had from you. We're constantly looking for new topics and would be happy to discuss something that you want us to cover. So again, we are thrilled to have you here, and we are looking forward to a fun episode. To kick us off for the podcast, for our 65th podcast, John is going to go over today's podcast topics a bit before we jump into the news and resource section. John?
Hi, so as usual, we're going to start with the news and resources, talking about COVID topics, then non-COVID, and anything else beyond that. And today's main topic is a discussion of early non-clinical bioanalysis, talking about non-GLP studies, GLP, and then some other expansion beyond that. Let's move to the news.
Great, John. Why don't you just kick us off? As always, we'll start with some some COVID news and then non-COVID news. And John, why don't we just jump right into what's the latest on COVID?
Well, okay. I think this is actually quite salient at the moment. It's actually the headline is Johns Hopkins winds down pioneering pandemic data tracking, and basically this was the Johns Hopkins University in Baltimore in the early days of the pandemic put together a website that was tracking cases and deaths, first of all in the US, and then it was international. And certainly for us, when we're talking about COVID, it was a really useful thing to refer to, particularly at the time where we had no vaccines, no treatments, and just watching the progression of the virus across the globe. And I mean, certainly, friends in other countries and things like that. It was, I would check on it fairly regularly.
Yeah.
But the stage now where basically their information sources are drying up, where all the states were reporting data, now it's really just down to a couple of places. I think it's like New York, Arkansas, and Puerto Rico, the only places that are reporting now for the US. So they've decided it's time. March 10th will be the last day where they'll publish, and then the website becomes it'll be archived, but still there for access for people to check, because it is, like I say, very, very interesting historically. And I think from our perspective, it sort of mirrors what we're seeing, where we're putting together, an agenda, and occasionally like COVID is a big splash, but really now sort of 3 years into the pandemic, it's like COVID news is really much less. But there's much less to talk about, and it's much less interesting than what's going on in medicine generally. So I think it makes sense for us to follow the Johns Hopkins path and sort of shelve the COVID piece to be just part of our general news topics rather than have its own section. And I will pass it back to you on that one, Dominic.
So many groundbreaking things. So we're going to change the structure of our podcast going forward, as well as John Hopkins winding down what was Time Magazine's one of the top 10, and I believe top 10, and I don't know if inventions is the right word, but top 100 inventions for 2020 was this website. And John, you're absolutely right. I mean, it ran for about a little over 3 years. This is in and of itself, if you're a student, it's got to be one of the most fascinating things, right? We kind of had, at least HIV was something that was I think of a pandemic, right, or an epidemic, right? And so that was where a lot of what I learned about viruses and virology was like spawned from that. And now I think there's a whole generation of scientists that are going to look back at COVID and really, it really allows you to understand just how the fine balance between like health and sickness is, right? And this was an unbelievable 3+ year run by Johns Hopkins. And John, you're right. In the early days, it was one of the most comforting. I don't know if comforting is the right word, but it was something to look at, to gauge, and gave information, and it settled me down personally, and it allowed me to, when people would ask me some questions, I'd go to that website and take a look at what was happening, and it was a very beneficial website just from I don't know, just kind of peace of mind. But overall, I think this is like a course, right? I could see it as like a graduate level course, the whole thing. So maybe I'll pass it back to you for some final thoughts. But for me, it's like, what a wonderful tool to be used to study, right? I mean, unbelievable.
Yeah, I'm sure there'll be publications, etc., based on that information as it sort of moves back more into the background. But yeah, I mean, look at the website was viewed more than 2.5 billion times.
Wow.
And generated more than 200 billion data requests. So I mean, it was incredibly valuable. But it's like, if you can't get the data in, then ultimately you can't get the data out, and that's really what it ran into. When obviously states of their attention has gone elsewhere, and so they're just not reporting this stuff. So let's move on to the non-COVID stuff, which, hopefully last time we'll say that. First article is decoy receptor protects CAR T cells from destruction, and this is one of the so CAR T therapy is obviously a big deal in terms of creating a immunotherapy from donor T cells, and it has had applications, but where it struggled is in solid tumors and things like that, and the biggest challenge is actually the patient's immune system attacking the CAR T cells. And this group from University of California, San Francisco, may have found a way to get around that by actually adding decoy receptors on the CAR T cells, which the antibodies preferentially bind to compared to other receptors in the cell. And so basically that kept the and this was a study done in mice, kept the immune system from destroying the donor cells in mice. And I'll get back to you because this is obviously close to your heart.
Yeah, I mean, this is an amazing breakthrough, right? So maybe we'll take a small step back. CAR T cells have known to develop antibody-mediated rejection, and it's a lot like graft versus host disease, only it's way more susceptible because an organ's kind of in a more I guess a less immune surveyed area versus a T cell is in your actually in your blood, right, where it's constantly going to be surveyed by the immune system. And so these scientists have figured out CD64 is an FC receptor, John. So CD32, 64, 128, very clever. They created them. It's FC receptor 1, 2, and 3 respectively. It's kind of easy to remember if you know that, right, John? So CD64 is, and when I say that it binds FC, that allows for the antibodies that are potentially autoimmune to bind in a non-immune configuration. So it's not going to kill the CAR T cell. So they've put up what they call them decoy, although I think it's a good way to represent it. So I hope that made sense, John, right? So you put up, and any antibodies are going to kind of bind to that CD64 because everything's Brownian movement in the body, right, John? It's not like it's got this sophisticated kind of guided missile system or anything. It's just all interactions and antibodies fall on and off, proteins fall on and off. Sure. Yep. And so if that CAR T cell now gets littered with antibodies in an FC receptor fashion, it putatively prevents it from being attacked in an autoimmune fashion. But somewhere in there's also something called antibody dependent cytotoxicity. So too many of them can kill the cell. So it's an interesting, like everything in the immune system, it has an ADCC type of response is what I described there. But everything in the immune system has a checks and balances in it, John. Mm-hmm. But it's a great breakthrough. I don't know if you have any questions about what I just said there, but these are the types of things that make me warm and fuzzy to be a scientist, because somebody like they just connected one and one and made two. But this is how we get to the next iteration of, off the shelf CAR T cells, John. That's what this
Exactly.
is.
Yeah. It's one of those ones for me. I've read this multiple times. I'm still digesting it, to be honest.
It helps to have that immunology degree to know that those are 32, 64, 128, all the same pathways, or they bind slightly different FCs to them. But anyhow, yeah.
But it's still one of those things you can see even as a neophyte. This is something that could represent a big step forward in the long term as people build on this research.
Yeah, and I don't know if you're still you call yourself a neophyte, John, but you know enough to be dangerous these days. But I like the use of that word. But I agree. This is a critical step towards what we all covet, John. It'd be great to be able to walk into a hospital and get an allogenic CAR T cell to treat X disorder, right? Like, okay, hey, come back in, a month, and we'll have your CAR T cell ready for you with one visit. So that is the expense of that. It would reduce the cost of things. I mean, it's off the charts what this could potentially lead to.
Yeah, and it's interesting that we always think of CAR T as associated with oncology, but this is actually it's what they've done seems to work for other cell types. They're talking, conditions that affect the thyroid and pancreas, such as hypothyroidism, type 1 diabetes. Thyroid cells and other.
We do, although you and I don't, because a few years ago, rare diseases became probably about 2-3 years ago. Or maybe even 5, where rare disease became not just a business model for CAR T or for cell and gene therapies, but an obvious unmet need that these were going to satisfy. And so you're right. It isn't. This has potential for really the sickest and the those that have absolutely no therapies, nothing approved. This is where it could really have its biggest impact.
Yeah, so like I say, this is just proof of concept, in the hope that this leads other researchers to go in this direction. That actually makes the technique and the therapies cheaper and more accessible, like you said. So yeah, I thought that was it's we're always looking for the where are we going in the future, and this is
well, it might not be for you and I, John, but our grandchildren got a shot.
Exactly. Yeah, exactly. Everything takes time, and I'm not going to be around long enough to.
We're going to have the traditional CAR T cells. Take a piece of my rib if you need to generate something for me. But over time, the allogenic is the way to get everybody treated, which is obviously what it's all about.
Sure. Shall we move on?
Next one
is non-essential amino acid plays an essential role in neuropathy, and so this is this is much more basic actually, but still a very useful finding. So in the US, there are 37 million people with type 2 diabetes, and a lot of them suffer from peripheral neuropathy. So it's pain, numbness, and weakness in the extremities, like the hands and feet. So scientists have long known that high levels of glucose circulating in the bloodstream can lead to nerve damage that causes neuropathy, but also they're finding that levels of some amino acids are playing a role too. And in this case, so the background to it is researchers from the Salk Institute showed that low levels of the amino acid serine put diabetic mice at risk of developing peripheral neuropathy. So now they're trying to develop a test that can indicate whether a patient could benefit from serine supplementation to basically alleviate those symptoms. So it would be interesting to know the background to that in terms of the difference in levels of serine, because obviously we have an amino acid panel that we routinely run. So it could be something we could do within our mass spec group if you're seeing enough of a change. But I just, for me, this is much more basic body chemistry, but still the implications of just one amino acid. The implications of that are actually huge for a large population of people.
Yeah, I mean it's a really interesting article, a very powerful finding, right? That serine could be such a major role in type 2 diabetes, right? And it's all lot of it relates back to sphingolipid pathways, right,
Yeah,
John?
sure.
That's not a strength of mine. I mean, I know
Yep.
Don Duffield would be a great person to be talking to this about, right? Like a world's expert in measuring these things. But it's certainly one that has profound, like you said, so basic, and we don't mean to underscore the word basic there, but
No, not at all.
it's such a simple solution. And now, no, it's a simple finding or a basic find, but it's really hard. It's not as simple as just dosing people with serine. That's not going to work, right? They're still not so sure that would be the best way to treat this. Instead, they're looking at oral glucose tolerance, which I don't quite fully understand all that, John, but it is definitely something that's what a major finding, as well as for what is it? A good percentage of the population has type 2 diabetes. I don't know what it is. It's very high. I feel like the last stat I saw was 30% or something. But this neuropathy, you hear a lot about it, and not just type 2 diabetes. I think it's senescence as well. And you wonder if there's a way to even treat some of that, because it's still an incredible unmet need. One of the downsides to living so long is that you end up with pain. And oftentimes these are the types of pain that manifest itself, numbness of feet and hands and things like that. So I don't know. I think this has like huge benefits throughout, although it looks like they're still in mice and there's still some stuff to be developed. That's why we love it, though, John. This is how it starts. These are papers turned into medicines.
Yeah, I mean, yes, absolutely, it's still in mice, but again, it's showing potential ways of attacking this, and certainly they say the actual scientists said, the data suggests that putting supplementation of serine can slow the progression of diabetic peripheral neuropathy. So maybe we could give supplements, but sadly, it's not as easy as that. I first passed through, I thought, oh yeah, just give serine, but they basically said that people have to take high doses of it to make a difference, and then also said not everyone needs more. So it's yet again, it's one of those things that they've established a pathway. They've certainly shown that serine is key to a lot of this, but then the actual how you address that is a whole nother research project in itself, and that's where the challenge is.
Yeah, it's almost reminiscent of like calcium, right? We thought that was going to fix everybody's bones back in, maybe 15, 20, maybe 25 years ago, 30 years ago. And for some people, it really does help, right? Some people take calcium and it can really help with but others, you can dose them with mgs of calcium. It's not going to affect them. It's an uptake, right? So like each disease has its own sort of unique kind of features to it. And it screams that, right? I don't know why I picked calcium, but it kind of falls right into that bucket where some people, maybe it is as easy as giving them a serine, a low serine pill or something, where others, it's not, it's got to be much more complex than just that. So interesting, John, and what a great couple articles on basic research here.
Just on that first piece, I mean, they basically said the Salk scientists are looking to develop a serine tolerance test similar to the oral glucose tolerance test to diagnose diabetes, to help then clinicians understand the patients that may benefit from serine supplementation, so there will be a subpopulation where that's the way to address it. I mean, even the subpopulation of 37 million could be a significant number of people.
There it is. That 37 million, that's not a small number.
Yeah, exactly. But even if you're 10% of that, that's almost 4 million people.
Yeah.
And then obviously you have the ability to figure out, well, if we've addressed that 10% or 20% or whatever, how do we then address the remaining population? But so when I say basic, I don't know that you like to say that was not casting any aspersions on it. It's really sort of fundamental in terms of it's low tech compared to the CAR-T stuff.
And it's still done in a basic research setting, John. That's what that versus like I wouldn't call what some of the major pharmaceutical companies are doing as basic research, although I bet you several of the people working there would tell you they're in basic research, right? So, you're right. We don't mean to. I think our audience appreciates that. We're not saying it's like basic as in fun, like simple, even though in theory it kind of is, but there's nothing basic about it. What a great discovery.
Yeah. So let's move on. And again, we're touching on all the hot topics today. This is potential preeclampsia treatment delivers mRNA to the placenta, and obviously there's a lot of discussion about mRNA in the media, but this is because RNA technology has been used obviously a lot in vaccines for COVID, and that's it's been technology that's been worked on for years, but that's really why it's become much more high profile. But this is researchers from University of Pennsylvania who actually are looking to use it to tackle preeclampsia, which is a condition. If you've had the kid, you're always aware that this is a potential issue. Where so basically, researchers from University of Pennsylvania are looking to look at how RNA can help address preeclampsia, which is a common but dangerous condition in pregnancy, where malformed blood vessels to the placenta prevent proper blood flow between the mother and fetus, jeopardizing the health of both. And certainly, if you've had, if you've had a kid, well, I haven't had it, my wife's had kids, you're very aware of that during pregnancy, and that's something doctors are always looking for, simply because, like the article says, it puts the lives of the mother and the child potentially at risk. So basically, this team looked at using lipid nanoparticles to deliver mRNA expressing a blood vessel expanding protein to placentas of mice. So again, we're back in mice, but again, it's showing it's proof of concept as to see if this can work or not.
It's a very dense topic, John. It's one hard to unpack, but
yeah.
I don't know if you want me to jump in here, if you had any last final.
So I'll just say, so it's a proof of concept, and part of the issue here is, as the article said, although you know about it as a potentially dangerous condition during pregnancy, there isn't really much understanding of it. So there's treatments, but again, there's nothing really universal for it. So they will possibly give hormones, minerals. The inflammation can be part of the underlying causes, but it also has the knock-on effect that if it occurs, babies are delivered prematurely, so that has other health complications. So this is it's a large population that could potentially be addressed by this. So I'm going to pass it back to you.
Yeah,
John.
well, John, that's something I just happen to know a little bit about from. They even mentioned in the article it's been known for about 100 years about preeclampsia, and they still don't have a good root cause, right?
Mm-hmm.
Like if you think of cervical cancer, it's like, well, the papillomavirus causes it. And even immuno-oncology, we always kind of go back to some sort of, here's what caused it. Preeclampsia is in that rare, I don't know about rare. I think it's in a disease state where it's still it's still somewhat unknown because it's so hard to treat it, right? So they think it might be due to, and when I say it's so hard to treat, not viable. Most moms are not going to want to sign on to an experimental vaccine of some sort while they're pregnant, John. Even if preeclampsia is a potential diagnosis that you have, because I would imagine a good percentage of preeclampsia goes as precautionary. Maybe they do some treating of symptoms and then the baby's delivered and it's fine. Although it can have, obviously, I believe it can cause stillborns and abortions and all sorts of things that are horrible for anyone to go through. But it's made it really hard to want to research it. And there's a book called The Cutter Experiment that showed that there's vaccines that can treat gram-positive infections are another thing that women can fall victim to, and it might even play into preeclampsia. So some sort of a standard infection could cause things, and there's vaccines that can treat that, but they've never made it to market, in part because it was heresy to want to treat a vaccinated pregnant woman, right? And so, sorry, it was heresy to treat a pregnant woman with a vaccine, and that's still a controversial topic. But I digress a little bit around how this is an age-old problem. So first, to the scientists at Pennsylvania, hats off for tackling what is the Mount Everest of treatments. This is not an easy space to be working in. What they've dialed it into is VEGF, and VEGF is vascular epithelial growth factor, a very potent thing in angiogenesis, John. So tumors produce a lot of, they make a lot of VEGF to increase their vascular intakes.
Mm-hmm.
And so that's how a tumor's grown. And so the Carl June went after anti-VEGF, and it's still treated in, that's a lot of what your eye treatments are to prevent certain eye diseases, anti-VEGF antibodies. And so you can suffocate the tumor by trying to, in theory, cut off those VEGF growth factors. And so this is a hot topic, and to see it transitioning into lipid nanoparticles is probably kind of a natural progression. There is a lot, you said it nicely, there's a lot of controversy around mRNAs, but despite even that controversy, I still think these have been given to people, not think, it's factual, for the better part of 25 years. Immuno-oncology patients have been getting mRNAs. So we know they have some safety profile to them, although I'm going to stop there. But I think this is something that it's still TBD. But I love that they're tackling it, John. I think that's the biggest. What I like the most about this kind of like post-COVID times is that we're getting back to people focusing on things that were really society detrimental to society, is how I think of it, and we're kind of back on that track. So this is just a wonderful story, John.
Let's go into some of the details of it. Like you say, it's all about restoring normal blood flow to the placenta by dilating the blood vessels, and as you mentioned, VEGF was key to that. So this group actually, with the lipid nanoparticles, that ability, where it goes depends on the structure of the lipids. So they basically looked at a number of different lipid nanoparticles and designed them based on ones that were optimal for delivery to the liver, because the liver and placenta have important similarities. So it says both are highly vascularized with high blood flow. So they designed a library of 15 LMPs, each using different lipids, and then added them to placental trophoblasts, a cell that's found in the placenta of both mice and humans, and tested how well they delivered mRNA with a glowing that's obviously the detector, with a glowing protein called luciferase, so they can actually obviously monitor how the delivery looks. Sorry about this. So they had one lipid nanoparticle work particularly well, and in terms of going to the placenta and also delivering its mRNA with higher efficiency than the others, without any of the payload finding its way into the fetus. So again, it's that key, not just delivering to the target, but making sure it doesn't go elsewhere. And so they did it then with VEGF mRNA, and they showed that yes, they could dilate placental blood vessels. Working in pregnant mice, they injected the VEGF mRNA into their tail veins and then analysed the placenta 48 hours later, and they found that the mRNA was able to dilate the blood vessels successfully, acting on not only the trophoblast but also endothelial cells and immune cells within the placenta. So it's basically they've done the foundational work, and now it's test this in mice with preeclampsia, and that's work that's ongoing. So you're right. It's addressing an age-old condition, applying modern technologies to help that, but really thinking carefully about the properties of the target, in this case organ, or organ in inverted commas, to actually get the delivery there to try and develop something effective. So this was something that just jumped off the page. It's like, we definitely have to talk about this, because this is really impressive stuff.
Yeah, no one wants to be faced with your own health or your baby's health, right? Like, hey, take the baby out early, and you could help improve your blood pressure, right? That's what this is really kind of having. Or keep the baby, and you might have something happen to you. Unbelievable decision makings there. I can't imagine. I'm sure as we sit here, somebody's having to deal with that, and the thought of being able to remedy it is something we definitely want to see. But back to a little bit, I'll touch you. There's a lot that's incredible about what they did in terms of luciferase allows them to image it and see it,
Yeah,
right?
sure.
So they can
Yeah.
kind of quantitate where it's going. And then to see how powerful the response was in terms of putting it in looks like they used the tail vein entry of the VEGF and saw that it acted on everything, which is really cool too. So it has a lot of just potential to it, and stay tuned. I hope in a few years we're still here doing this and we can learn more about that. But that's what a incredible advancement that would be to treat something like preeclampsia. It's wonderful. It's making me warm inside to think that we're getting back to normal. Speaking of normal, John, right? I think that's good for the news, right?
Yep, yep, absolutely.
We're ready to go on to our next topic, which is really becoming I'm going to call it a hot topic. I think everything's a hot topic, but early non-clinical bioanalysis has certainly changed a lot in the last couple of years, John. I mean, it's coming full circle. It's a fascinating space, one that certainly I'll kind of jump right in, John. In the biologic space, it's been I'll say, I don't know if controversial, but somewhat like, why are we investing all of this in these non-clinical studies when it really may not translate into clinical, or even worse, it's not, I guess, not only not translate, but it's really not helping at all. So
Yeah,
yeah, and maybe we'll just take maybe I don't know where you want to start with, but I think our first topic is there a need for animal-based studies and intentions? And I'll kick it back over to you because I definitely have a lot of thoughts about this.
I'm going to be brief on this piece. I think it's very interesting that we last time we talked about the FDA ruling that they want to encourage more use of non-animal models um, in terms of early research, and obviously that will have its own challenges for industry. And I remember us both talking on the podcast saying that's got to be a good thing to minimize animal usage, etc. But there's still going to be conditions where you still need that animal piece. So there's like a balance that we're trying to strike here. But then every news item that we've talked to, with the exception of the Johns Hopkins site, closing today, is all talking about what we've done in mice to then for the long term, hopefully address conditions that are of importance to human health. So it's really underpinning how valuable animals can be, and certainly that all the stuff we talked about is proof of concepts. Is very, very early stuff, but it's amazing how, when we decided this was our first topic, then how we all the news was really saying how mice, which are not that similar to us, but really are incredibly important to medical breakthroughs that could drastically help
Yeah,
human
yeah, certainly, John. Yeah, I couldn't agree more. There are times when we're not here to say, hey, let's eliminate all this. That's
no,
You
no,
can't
no.
go from like relying on the model of one, kind of rodent species, one more advanced or larger animal before you go into the human and just abandon it abruptly. That's crazy, right? But to your point, maybe there's ways to look at historically, what do we know about a small molecule and a biologic?
Yeah.
Maybe we can start by reducing the number that we're
Yep.
doing. We're going to
Yep.
talk more about that in a minute here. But high level, there are many groups that are looking at eliminating this type of like animal-based studies, and one is PETA, right?
Mhm.
And there are some others, and those groups, what they would like us to move towards is it's just more in vitro studies, John. Right? We could
Mhm.
take human cell lines from, say, cardiac tissue or kidney tissues or kidney cell lines or lung cell lines, and we could evaluate all different types of cell lines in vitro and get some assessment and glean information. But that has to become put into practice, and to me, studied in parallel with the current non-clinical work. And then we have to start to understand, what is that information telling us, right? How is that going to guide us? Because right now we could do it. That's not the problem. And they do. And
Yeah.
certainly in CAR T cell settings, or any cell and gene therapy, oftentimes they'll screen hundreds of cell lines, or not hundreds, maybe 25, upwards of 50 different cell lines across every tissue, right? Bladder, pancreas, just to make sure there's no in vitro toxicity effects. But that's a major investment right now, John.
Yep.
And so how would we be able to streamline that? How would we be able to replace it is where that's the high level, that's the crossroad I see us at today. And I'm going to maybe, there's elements of things I think we're already reducing in the non-clinical space. That without a doubt, we know, for example, the amount of investment in the amount of animals for immunogenicity of biologics is being reduced significantly in the non-clinical space.
And
But the next step of just eliminating it, we're not quite there yet, John. There's still a lot of powerful. Trust me, there's still many a drug that they get 4 or 5 different versions of it, put them into an animal, and it may cause a lot of toxicity. We got to still keep some of
sure.
that until we have
Yeah.
a surrogate for it, right?
Yeah, and I think that's where I said about intention. I mean, really, for us right now, and you're right. It's long term. Maybe we can move towards phasing out animals, but we're certainly not there. It's more about the reduction, but the intention really is to figure out what is your investigative drug going to do in a biological system. So it's looking at the pharmacokinetics. So how high a concentration is it reaching, how long is it hanging around, how does it affect other biomarkers and pathways in the body, and then is there a toxicity effect? I mean, for small molecules, we're talking metabolites. Is there the actual drug of interest might be non-toxic, but is there a metabolite that actually could cause major problems downstream? I think for us, in terms of reduction, bioanalytical can help with that. It's we obviously the progression we've seen at least on the LCMS side of things is you get that instruments have got progressively more sensitive. So there's things that we can reach detection limits or lower limits of quantitation that we could only dream about, years ago. Because the instruments are getting more and more sensitive. So, but that also can be helped that we don't always need high sensitivity for every analyte, but because we've got a more sensitive instrument, we can extract from smaller aliquots. So by being able to do that, then for things like mouse or rat studies, rather than having multiple rats or mice you've got random sampling, you can actually take it from one mouse, but or rat or mouse, take much smaller volumes, do serial sampling, and there's an immediate reduction in your animal usage. So things like that are, bioanalytical can definitely help on that front.
Yeah, I mean, I think well said, right? So there's 2 major reasons why you do it, right? The pharmacology of it and the safety or the toxicity.
Yeah.
I'm for biologics, I'm definitely more focused on that toxicity component. Not that the pharmacology isn't critical. It's just, there's small molecules tend to translate a lot better in a
Yep.
pharmacological model from non-clinical or animal studies, right? Where biologics, it has a looser, still has some value pharmacologically, but not as great. And there is no real metabolite generation. That in and of itself is something that maybe hard, or it's hard for me to envision eliminating animal studies, because there are many I believe you can use liver cells or kidney cell lines and things like that to try to look at those metabolites that might be generated, and some of it's very valuable. It can be verified in an animal, but I don't think it can replace it to date, right? And maybe there's some AI, and we got so many advancements. We got to, the industry's got to come together. The first step is just talking about it and movements, right? This is stuff that's again, I don't know, as recently as a couple of years ago, John, you and I would have said, "Oh, there's no way we're eliminating this stuff." Right now, we're much more in tune to say, "Hey, or we'd say, "Hey, great, we'd love to eliminate it, but how?" Right?
Yeah, sure.
And now I think there is more arguments towards, maybe not eliminating, but certainly reducing it over the next 10 years. You could see us cutting this by a fifth, right, or a third, or something dramatic, which is really what it's about. It's just nobody wants to unnecessarily perform non-clinical studies, John.
Yeah. I mean, I think it comes down to, in vitro can tell you some information, but does the animal model tell you more? And if it does, then that's going to mean you're more likely to work with animals, to get the information you need.
Yeah. Interesting. John, what do you think? We can go on to our. So I hope that set the ground for
Yeah,
our
sure. Yep.
audience here around our topics. Not the need, but early non-clinical bioanalysis and just animal studies. And if I hope everybody understands how and what that is, and if we want to drill a little bit deeper, John, it's like, we kind of touched on this, but the different needs in non-GLP analysis, right?
Yeah, let's go to
Um,
the next.
I think
Yeah.
that's a good one, meaning like how much do you have to invest?
Yep.
How accurate and precise does it need to be? How much do sometimes we put a couple $100,000 into a method that is super precise, super sensitive, but the reality is we didn't need all that, right,
Yes.
John?
Yeah, go in that direction. Yeah, let's kick off that. Yeah.
Yeah. I mean, you can if you want. Why don't you jump up, because it's very different. I feel like pharmaceuticals and biopharmaceuticals have different needs when it comes to these things, but certainly in our world, there is more and more people moving towards non-GLP for the PK component of things. Now, if you're in a full GLP tox, it still has to be GLP, but clearly on the immunogenicity side, depending upon the immunogenicity risk profile of the drug, and things like monoclonal antibodies are really low. They're low risk drugs. As
Mhm.
recently as 18 months, 24 months ago, you did a GLP tox. You just validated all 3 tiers of the ADA assay, and that was it. Now we're moving towards a non-regulated way of doing it, and you look at either just a couple tiers and calling it good, and that's dramatically reducing the cost. I don't know if it really reduces the number of animals, but that's one step towards what we're talking about. If we can start to find alternative ways in reduction, that leads to an eventual off ramp onto different venues instead of animal based. But the immunogenicity, I just wanted to touch on that real fast, because that's a slightly different sort of component versus the PK. But if we jump back into the PK, John, I'll kick it over to you, and you can
Sure. Yeah.
start drilling in there.
So one of the things that we talk about is the different needs in non-GLP analysis, and I think from my perspective, and you almost have to rein yourself in as a bioanalytical scientist. We're always thinking we want the assay to be as good as possible, and it doesn't always need to be. And when you're talking, some of it is you really have to listen more to the customer, because even in a non-GLP setting, we know we're not validating, but we're going to come up with an assay, and we want to qualify, because we want, as a bioanalytical scientist, our instinct is to say we want to show this assay is ready to do your samples, and by qualifying, we're showing that it's suitable for measuring samples, because we always think that your samples are the most precious thing here, but in a lot of cases that isn't necessary. It's really all they want. All the customer wants is a number that is relative compared to other test compounds, things like that, and it's just the degree of difference might be good enough for them to make a decision to move forward. So that so that again plays into how much do you need for a sample, is to help guide the animal sampling, because if your assay is on the edge of in qualification, you might not need that, and you might think I need a larger aliquot to then make this a more robust assay. So some of it is education in ourselves, and so you really have to be open to thinking that as basic as possible will work here to give the information to then move things forward, and hopefully minimize the amount of work that needs to be done.
Yeah, John. And so I think I got us a little sideways, right? There's a non-GLP component and then the GLP component.
Yep.
And that non-GLP component is where we're drilling in right now. And that is an area where typically you may have to pool animals, right, John? You have to do all sorts of things that, if I understand you correctly, we may be able to reduce all of that by having a method that doesn't require a larger volume, right?
Yeah.
And that becomes a major savings of the number of animals you need. This isn't all about money, by the way. We're really talking about the animals themselves. We could significantly reduce the number if, for instance, you don't need a non-GLP method that requires 200 microliters of sample, right, John?
Sure.
And that typically it requires that to hit the sensitivity levels that you need in order to get a true quantitation at the lower limits and generate a good PK profile, right? So then it becomes, can we get away with doing less? I think that's where the non-GLP. Do I have that right? Is that
Yeah, I think that's where I think
yeah.
we can help here. Yeah, yeah.
Yeah, and that's a huge. It's not a small number that we're talking about in terms of reducing, for instance, maybe the need for 3 groups times 6 to just 6 animals, right, John? You could cut it down
Yeah,
by a
yeah,
third, and
yeah.
that's when you go across all the drugs that are in the discovery state, that's not a small number there. So that's a wonderful way to really kind of drill into that first step towards where the field's going. It's how robust does the non-GLP analysis need to be? And we've always talked in that we don't like these terms, right? Like quick and dirty, John. We don't like that. But what maybe I don't know if you want to share a little bit about like how that works, right? You might not have a standard curve, or if you do, you might not have it in matrix, right? Those are all the things that are some of the savings, right? That can be done, and not financial savings. I'm talking about the matrix in the animal, right?
And that's an interesting point. I mean, there is that guide. There is sort of ongoing guidance to sort of possibly look at less use of biological matrix for preparation of curves and things, and using the surrogate approach because it minimizes use of control plasma, which comes from a central animal facility. But again, it's less sampling of the animals for that helps the downstream in terms of number of animals used. I mean, certainly, EBF have a lot of their focus going forward is like how, as bioanalytical scientists, can we be more responsible in terms of consumption of things like that. I think we'll hear a lot more from EBF on that front.
Yeah, I mean, I almost want to play devil's advocate here, right? Like, hey, we're all in, but we don't want to compromise the safety. That's the piece where I'm not struggling. It's just we have to use some caution here, right?
But again, it's like there's levels of non-GLP where even if you compromise, it probably doesn't impact the conclusions that people want to make from the data.
Yeah, no, that's very true. I mean, really, most of the time they're just looking to get did the animal not, like, spontaneously die, right? and then, does it look kind of how we expected within, I don't know, maybe 30%, 50% of what we were looking for. So it doesn't have to have this.
And did it look better than the other therapy that we were testing?
Yeah.
I mean, it's like I say, it's comparative rather than absolute. And like I say, as an analyst, I always want as good a data as possible, but you have to sort of educate yourself on that. Anyway,
yeah, that's the change is hard though, right? You just touched on it, John. You said it very well right there. It's like we want the best.
Yes.
Yeah. Maybe it's a good segue off of the non-GLP into maybe, do we want to talk about biomarkers in general and how, or is that a good?
Absolutely. Yeah. Yep. Go there.
Yeah. No, I mean, I think this is I've always been a big supporter. So part of the challenge here when we talk about non-clinical bioanalysis is the modality of the drug, right, John? So already some modalities, biomarker and even pharmacodynamic testing, is heavily relied upon in the non-clinical setting, right? And then other modalities, it's almost like, well, why would I do that? Because it doesn't really mean anything. So it's got this, I feel like, really a couple different landscapes there. But traditionally, small molecules and biologics, it's been somewhat of a minimal amount. Maybe you'd invest in a biomarker that is, like, I would say, really germane to your mechanism of action, a known one, that a specialized one. But the idea of screening pro-inflammatory panels is, well, yeah, of course, you're going to put a humanized antibody into a mouse, you're going to generate some pro-inflammatory cytokines, right, John? Why would I look at that?
Yeah, sure.
And then, of course, in cell and gene therapies, it's way different, right? That, I think you got to look at that, because it becomes especially when you look at certain gene therapies with the exposure studies, and you're looking at biodistribution of the viral vector, for example, and all of that becomes a much more heavier investment in the I think of it as a biomarker side, because you can't take the pancreas out of a human and take a look at it, right? So this is some stuff that becomes a little bit hard at this stage to think about. I touched on those cell lines as a potential remedy, but I think we'd all agree before it goes first in humans, we would certainly want to be looking at it in some evolved animals. But
Exactly. Yes.
yeah.
Yes. Yes.
No, I mean, I don't know if you have anything to add on biomarkers, because I think a lot of people I think there's some misperceptions there. They just think, oh, you could just use biomarkers. It's like, well, wait a minute, right? That's not going to tell us the full picture.
Sure.
And,
Yeah.
and maybe even then you're actually biomarkers in a setting you may actually have to even use more animals, is what I'm saying. In the cell and gene therapy space, some of those trials have to use more animals because they're trying to figure out if this viral vector is doing two things. Does the viral vector itself going off they call it off target, right? Is it going to a tissue type that it's not supposed to be treating, right? And then is it virulent, right? So there's kind of two things. If not only is it potentially depositing itself in tissue that it's not supposed to, if it's there, is it actually is it becoming active? Meaning, is the virus aggressing? Is it growing? And that's like a viral shedding. So there's a lot of words that that I think scientists need to be very careful when they're choosing their words, talking in this space. And I'd like to see, on a side note, I'd really like to see like the thesaurus of cell and gene therapy for bioanalysis. I think I'm working on that, John, right? Like that would be a real handy thing if we could pull that up so that we all got on the same page. But I digress a little bit. So, John, maybe some thoughts on non-clinical biomarker analysis.
Yeah, I think obviously part of the reason for doing this is our final piece of this talk, and we'll touch on that when we get there. But ultimately, with non-clinical bioanalysis, this is something where we have a lot of expertise across all kinds of technologies. So we have two LCMS teams looking at smaller and intact molecules. We've got the hybrid LCMS team who do a lot of non-clinical work, where they're looking at digesting things like antibodies, large proteins, etc. And obviously, there's a ton of experience, both in biological fluids and tissues. And it's so we are there to provide services. We obviously want to be part of the discussion about how we can help drive less animal usage, etc. But ultimately, we're bioanalytical people, and this is we can provide value on this. In terms of once we move to GLP studies, we've got regulated teams who will validate, and that's where you're going to be doing much more rigid approaches to the studies. So your animal usage is going to be defined by the regulations. So because we know there's a wide raft of studies that have to be done to support applications to the FDA before we can move into the clinical piece. So that's where, so certainly non-GLP, I think, is where there can be a lot of discussion about what can be done to get the information that will get you to GLP. But once you get to GLP, hands are much more tied.
Yeah. And well said, John. I think that's a nice transition into our final sort of agenda item, and that is the true, progression into the GLP studies, where today, I like what you just said, our hands are somewhat tied. I touched on a little bit earlier in the immunogenicity space. We're going to shelve that because it's almost its own topic, right, John? We could take that as its own topic, but right now there is more leniency in GLP studies for the immunogenicity component of things. And when I say leniency, I want to be very careful. If you have something that's a mid to high risk, you probably still need to GLP validate your methods today. But if you're in that low risk category, I do know of a couple instances where people have had non-GLP tier 1 and 2 methods that have been, gone through the review and have been accepted, right? So there's still, I want to put some caution there to the audience, but that's the first step towards some of what we're talking about today, towards reducing the need for certain animal speed, reducing the number of animals in non-clinical studies. But back to today and the progression for GLP studies, John, we know it's not negotiable today, John, is it? It's just not. When it comes to the PK component of things, and we've had some people who have, certainly some of our clients, because of, when I say it's non-negotiable, I think what I mean by that is you have to exhaust a lot of resources to get to a point where you could justify a non-GLP PK assay for support of a tox study. And they can happen, but when I say that, that means you've looked at either you're treating something that's in an area that is not going to get into the periphery, and so for by default your sensitivity levels are so low that they're almost negative. And, right, John? I think that's an area where it becomes very challenging for people to develop a method that's going to meet the regulations if it's all
Sure.
negative responses, negative
Yep. Yep.
results. Your compound's so complex that we can't get an antibody on it. We can't, it's littered with things that just prevent it from being seen to help
Hmm.
keep it, stabilized. And maybe we try, some molecules might even be pegylated, but that's got some configurations, and peg antibodies don't bind well. And so the list goes on, and you might try 3-6 platforms and can't actually measure it, knowing that it's not going to be there. Wow, my head hurts, right, John? But
Yeah.
that's the exception, not the norm, John. That's like the 1 in 10,000 that's going to be in that category.
Exactly. Yes. 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 I mean, yeah. Certainly, we dealt with drugs where they're designed with a particular mechanism, shouldn't be in the blood, but you still have to try and develop an assay to show that you can measure it in the blood, even though the drug shouldn't be there. Yes, you're right.
Yeah.
That's the head.
But otherwise, it's non-negotiable, and you can't cut a corner. I mean, there's no like, oh well, we think the metabolite might be this. No, you got to go find that out, right, John? At that stage, you got to go back to non-GLP. It's almost like you got to go back to go, right?
Sure.
You don't get, no. To quote, I'm going to date myself, John. Do people still play Monopoly? I don't know, but you go straight to jail and do not pass go in those settings. And I don't mean jail. I should be careful. No one's going to get arrested, but John, it is in my mind, when we talk to clients, it's not negotiable if you can do a GLP study and your PK is non-GLP. It's just really bad practice there.
Sure. Exactly. Yes. Yes.
And
Um, and so, yeah, I suppose we talked about GLP. One thing we can touch on briefly is also dose formulations where it's out of the animal, and it's really again, it's key with dose formulation analysis, which we routinely support. But it's key to have that, get talking to the team as soon as possible to make sure that assays are there, and ready and developed and validated ahead of the study, such that you're not going to make so that no problems are observed in terms of dose formulation in a study that requires a repeat of a study, and there's increased animal consumption. Planning becomes key for something like dose formulation. Although you think it's out of the animal, it's in suspension / solution. But if you don't do it early enough, it can cause downstream issues, that then impact, exactly the thing that we're
what do
talking
you
about here.
mean, John? That never happens. What are you talking about? Well, no one makes a
Mm,
mistake with dosing the animal and results in funky PK profiles that bioanalytical labs have to deal with. No way, John. That never happens. No, we've never had some long nights and lost sleep, and it's part of the reason why I'm bald. No, 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 no. That never happens, John. But no, I say that in jest, but like
Our
there's one
soapboxes are
take-home
getting some
message
heavy
here.
wear today.
What's that?
Our soapboxes are getting some heavy wear today.
Yeah. I'll like put my little hat on and I'll get up and pontificate for you, right? But no, the I jest, but you kind of we scratched on it, but it's a progression, isn't it? Where you start everything in that non-GLP setting, from dose form to the PK assay to the immunogenicity assay if you're in the biologic space, and you do that early and often and learn as much as possible, so that as the method moves into that GLP state, you're ready, John. Right?
Sure. Yeah.
And it
Yeah.
doesn't, dose form is a key piece of it, and I don't want to take that lightly, because that can be really problematic if you don't have a nice method there. And
Exactly. Yeah. Yeah.
and one that is often over, like, the days of people overlooking bioanalysis are hopefully behind us, but it still rears its head. Most people are now aware that as they're getting into their CMC type space and they're making drugs in a pilot setting that are going to go up into their large scale GMP, they're proactively working in these methods earlier. That's a nice progression for us, right, John? Like 10 years ago, it wasn't that way. It was almost an afterthought to do bioanalysis sometime.
Sure.
So that's
Yeah.
a nice movement in the field. Obviously, we'd like to do more if we could, but I think it's really important that if free consulting to anybody listening, I'd invest heavily in my non-GLP stuff, John. I think that is really an important piece because it sets you up. Now, some might say, but Dom, if it doesn't move this and that, but if you've got some candidates that you think are really, you believe in them and they show efficacy, it's not a huge investment that I'm talking about. It's
Sure.
probably more time even than money when it comes to trying to build a more robust dose formulation, a robust non-GLP method, or at least one that sets itself up for a GLP analysis. And what I mean by that is often in the monoclonal antibody space, even the ADC space, you can get away with using commercially available reagents to support your non-clinical and even to some degree the GLP, the non-GLP piece as well as the GLP piece, right, John? But that has some real benefits for financially. It's helpful, and what I mean by that is making a monoclonal, an anti-ID antibody, or making polyclonal sera against it, so you have a building block for bioanalysis. And often that's skipped, and I get it, but those that invest in that have a much easier path when they
Sure.
move into the GLP, because that method now, what you're doing non-GLP can actually translate into a GLP method. In the biologic space, you use a commercially available antibody for a mAb that does not translate. You're taking advantage of your human antibody in a non-human species. The moment you put it into a human, all that you've invested in your non-GLP work goes bye-bye. So
Sure.
that's one of the strategies that
Yep.
people
Yep.
take advantage of, and I get it. But to me, I'm going to spend the 30 grand and create some polyclonal sera at a minimum. At a
Sure.
minimum, I want that because
Yep.
it's a great. You can still use that as a capture antibody and as a detection antibody. It's far superior, in my opinion, than some commercially available monoclonal antibody against your drug that we're trying to squeeze. So they made this. They didn't. Whoever generated that didn't generate it for PK analysis. You're making partially purified polyclonal sera or a mAb that's specific for your needs. I mean, just you can hear my passion getting out of me, John. Right now, I'm really getting on the soapbox, but I'm not. I don't want to willy nilly say, "Oh, you should invest in this," but man, it could be a huge advantage, time saving, all of it if you pick that investment in the non GLP space.
Absolutely. Yeah, I totally agree with you.
And then maybe we'll just end with a shameless plug here, John. Right?
Yes,
Like
absolutely.
yeah. Go ahead. You tell us about it because I'm super excited. I'm going to be going to this.
Sure. Yeah. So basically, when we back and forward ideas of what we're going to talk about, sometimes there's a reason behind it. Other times it's a little bit, oh, we really need to talk about something. What we're going to talk about. This one, there's definitely a reason behind picking early non-clinical bioanalysis, and that's because a number of scientists from KCAS will be at the Society of Toxicology meeting in Nashville in March 19th to 23rd. This month in sort of 2.5 weeks' time. So I'm going to pass that to you, Dominic, seeing that you're one of our representatives.
So it's I think it's the 86th Society of Toxicology, SOT. I have been one other time. It's been maybe 8 years ago, I think I went. It was in New Orleans. Big conference, John. Really very large conference. Super excited to go. Obviously, it's heavily in the space that we just talked about, meaning this is obviously, as the name says, Society of Toxicology. There's a lot of in-lifes there. There's a lot of people who have expertise in how to dose an animal with a certain modality. Some of these things become very sophisticated in terms of trying to give a cell and gene therapy versus a small molecule versus a biologic. The entry lipid nanoparticles are all the rage as well, and how to dose them. And we even heard some of it in our news and notes. But that'll be a huge section of it. It's that in-life piece of it. And then on the flip side, it's the bioanalysis of it. And that's a real big chunk of it as well. And that's right in our wheelhouse in terms of, the pharmacokinetics of it or pharmacology of the drugs. And there's a lot of discussions around the new modalities and how to handle some of that, as well as the toxicity by definition of the meeting itself. That's going to be a huge discussion area. So it's a meeting that has a lot of in life labs, as I said, still a lot of heavy in the CMC space. There is going to be a lot of GMP, and that leads nicely into that dose formulation. So there is a lot of talks around that because you've got two pieces, two kind of groups. You're in, and the in-life and the CMC are rarely the same. They can be, but typically they're completely different entities, but both have to deal with that dose form piece at a very high level. And then we pick that up, as well as the bioanalysis, and this meeting has some focus on that as well. So I hope I kind of set the tone there, John, but it's a great meeting. It's big. It's got a lot of I know I've already got a number of meetings set up. It's got a lot of people that we work with that'll be there. It's not the GLP analysis, the bioanalysis isn't always a focus point, but this year it is. And I even think there's a major push for trying to reduce the number of animals that we're using. And I do think there might be some discussions on movements to try to not eliminate it, but just, we got to keep talking about it, John, in order for it to happen. We can't just magically hope it's going to happen. It has to have compromise. It has to have good thought process. It has to have good trial and error to it before we can eliminate the use of animals, which I don't know if that'll happen in our lifetime, John, but certainly one that we all want to try to move towards. So super excited to go, John. I'm going to be driving. It's only like an 8-hour drive from Kansas City. It'll be a lot of fun. And meet me in Nashville if you're coming to SOT.
So I know a number of our in-life partners will have booths. Are we having a booth, or we
have a booth? Oh yeah, sorry, we do have a booth. Okay. We do have a couple. We don't have a poster per se, but we have a couple presentations specifically focused on dose formulation. So our director of dose formulations, David Masters-Moore, he's been doing this for 20 years. I would consider him a world's expert in dose formulation for sure. He'll be doing some booth presentations on Tuesday and Wednesday, and we can get those dates posted. But I think it's Tuesday the 20th, and Tuesday the 21st, and Wednesday the 22nd are the days that we'll have booth presentations on dose formulation. So he'll give I think it's like a 15-minute presentation at the booth on how we solve dose formulation problems on Tuesday the 21st, and then on Wednesday the 22nd. So stay tuned. And I don't know our booth number offhand, which I should know. Unfortunately, I don't know it, but we will certainly post that on social media here as part of our release of this podcast itself. So super excited, John. Yeah, really excited to go.
Who else is attending it?
Me. We'll have our senior vice president of commercial, Jeff Goddard. Our director of dose formulation, David Masters-Moore. And then inside business development, Sean Utley will be joining me. And Sean is a great business development individual who's thirsty to learn more about this area as well. And he's been tapped to help us. So we'll have a booth, a nice 10x10 booth. Like I said, we'll have a couple presentations on dose formulation. And then obviously I'll be there to talk as much as possible about some of what we went over today, which is no small part of the drug development, but certainly anything related to biologics, cell and gene therapies. And John, I'll be around to talk small molecules if needed. I know a little bit there too, but it's really last time I went, it was like a carnival-like atmosphere. I hope we return to that. I think we're coming out of that COVID. It is an all in-person meeting. I do think the number of attendees is several thousand. I don't know the final number, but last time I went, it was like 8,000 or 9,000 were there. And I think this falls into that level of attendance.
That should be good. Yes.
Yeah.
Okay. Well, in that case, I think we've covered the non-GLP. Unless you have any final thoughts.
No, it was a really fun episode, as they always are, John. I truly enjoy working with you on these things. I don't have anything else unless we want to touch on like our weekend or something.
We can. I can touch my weekend. It was very low key. We had we saw in the weather forecast on the Friday that we had this massive snowstorm coming, and as we went to walk to go for dinner, we had fairly aggressive snow coming down. But it actually just sort of fizzled out around 9 o'clock. And because we thought we were going to have a snowstorm, we were in the mindset we're just going to have a nice, easy weekend and not do very much. And, well, the snow didn't happen, but I still had the nice, easy weekend of not doing very much. So,
isn't that funny how it works that way? So Snowmageddon didn't come. Is that what happened?
Pretty much, yes, exactly. So just had a quiet, relaxed weekend. but I could do it another couple of days.
We were talking before we got on the air here, I had the pleasure of going to a lacrosse tournament out in St. Louis. So I drove across Missouri, which is a fun drive, and I know that's near and dear to Jeremy. We made fun counting the number of billboards we saw for certain things like I won't share too much, but it's fun to count billboards. There's a lot of billboards on I-70 between the four. It's about 3.5 hours to go from Kansas City to St. Louis on I-70, and it's like a straight shot. It's a real easy drive. But my son, he's junior, and he's playing on a varsity lacrosse team, and so they played a couple games. They lost by 2 goals and lost by 3 in the second game. So they played some tough teams. But more importantly, my wife and I had a blast in the car, and just an all-around fun weekend to go and do something adventurous. But it, like you said, John, it went by a whirlwind, and that was. I don't have any good pizza stories or anything like that. I did, nothing. Didn't even make anything good to eat recently, and so I can't go through a podcast without talking about food in some capacity, John. That would just not be right? So, I don't know if you got any parting thoughts, and certainly I'm. Maybe look proactively, I'm going to go visit Mickey right before I'll be on vacation starting Friday of this week. I'll be down in Orlando for 5 days. My 17-year-old wanted to make another visit down there, and we should have a lot of fun. I think we're going to Animal Kingdom, Magic Kingdom, and then one of the Universals, a day at the beach or two as well. But it's really warm down in Florida. Really looking forward to some time off in general, John. But so I do have next podcast. I can share some stories about not only my time in Orlando, but more importantly, right as we should be done with the SOT meeting so that we can share some of what we learned at the Society of Toxicology meeting. So got a busy couple weeks coming up, John, relative to what I normally do. A couple big trips in there.
So while we were talking, my wife was saying we really need to have a vacation, and I had to remind her we just had one.
Yeah, I was going to say, weren't you in Lisbon?
Yeah, which was like between the last podcast and this. Yeah, we went to Lisbon for a few days, which was honestly a fantastic city. We had food was great. It just had a really lovely laid back atmosphere, and now both of us can't wait to go back again sometime.
On my bucket list. Yeah, I've always talked about one. I spent some time in Spain when I was in high school, and just could tell Portugal was beautiful, right? Yeah.
We were only there a few days, but one of the key drivers to get us back there is actually one of my wife's brothers has like a small farm north of Lisbon. He came and met us on one of the days we were there, but they're sort of, I think when they bought the farm, it was like he and his partner, it was like a bit of a shell, so they're slowly building up to where they can have guests come and stay. So we're basically, now we've done Lisbon, we're sort of saying we want to come back, so let us know as soon as possible when you're ready to accommodate guests.
I'm still like, he could have picked anywhere. My son, he picked Orlando. I was like, you sure you don't want to like go to Germany or something, or Paris? Or what? He's like, no, Dad, this will be a good time. We can go to that when I'm in college. Must be nice to be able to talk that way. I know when I was growing up, it was like if I got a weekend at a friend's house in Cape Cod or a weekend in Vermont, I thought that was the greatest thing in the world. But the thought of going overseas has really got me. It's in my bones, John. I feel like I want to travel more. I'm a little envious, a little jealous, not envious. I'm jealous of you making your way to Lisbon. I think that would be fantastic.
The travel piece, every so often we go for like a while where we don't think about traveling, and all of a sudden you get this urge, you really like, I really want to go somewhere. And actually the next thing for us is, it won't involve me, but my wife is going back to the UK, and for her it'll be the first time she's been back since 2019, because obviously with COVID and everything closing everything down, so it's going to be interesting to see how that is for her.
Yeah, no, I mean that sounds awesome, and I'm sure at some point may make a trip over just to visit our Lyon site. I've been talking about that. Yeah. I think that
we all want to do that.
Yeah, I don't know if it's a want. I know it is a want. For me, it's right now my life will be consumed with lacrosse for the next 2 months. If you haven't picked up on that, he's got 2 games this week too. Like Wednesday night and Friday night. It's a whirlwind of a next like 8 to 10 weeks minus the spring break week. Obviously, there's nothing that week, but it's a lot of fun, and his team's good, and it's actually a club sport, and so parents have to help out, and so I find myself with a second job, John, is what I'm trying to say.
Yep.
And but I do it all. I do anything for him, obviously, and it's great. So no, John, that sounds good. I think we can. I talked about pizza a little bit, so I kind of got that in for all my pizza lovers there. I do have one little food thing. I got this tomahawk chop that I'm going to make. It's this massive steak with the big bone in it. I don't know if you never heard about these. They're like tomahawk, they're called. Yeah. I got one of them in my freezer that I kind of saw as I was grabbing some chicken for the week, and thought, man, I'm going away this week, but that thing needs to be cooked on my big green egg at some point, John. Sure. I miss cooking on that. I haven't cooked on it in like 2 weeks. I feel like I'm kind of missing an appendage almost. That's how much I like that thing. But anyway, making myself hungry, John. How about you? That's all I got. That's about all I've got. Although I do have big aspirations for my Uni pizza oven when the weather, it's kind of getting in that area where I'm going to try to master my Uni pizza grill this summer. That's
sure. Yeah, we were cooking yesterday, and both our ovens at different times gave out alarm messages, so I'm not sure what's happening.
What does that mean?
Yeah, well, one of them just refused to stop. One of them just refused to work. The other one, we figured out what was going on.
And didn't you have this massive issue trying to get it installed, right? Wasn't there?
Yeah, oh, absolutely. Yeah. Anyway, I think it is time to wrap up because we have to move on to other things. But I think we should mention next recording, we actually do have a topic already. We'll be talking about recent advances in hybrid LCMS, and we'll have a couple of guests. We're going to have Don Duffield, who heads our hybrid LCMS team, and then we have another special guest external to KCAS, and we will introduce them on the day.
Yeah, exciting. We love external guests, John. I like the way you left it as a mystery, and take care, everyone. We'll see you next time.